This presentation is about the interplay between hydrophobicity and the positive inside rule in determining membrane protein topology. Background. The energetics of membrane-protein interactions determine structure and protein topology. Hydrophobicity drives the insertion of helical segments into the membrane, and positive charge orients the protein with respect to the membrane plane according to the positive inside rule. In this article, authors present a topology prediction algorithm called topgraph based on a sequence search of minimum insertion energy. Topgraph use deep sequencing and in vitro selection of segments inserted into the bacterial plasma membrane to infer insertion energy profiles for each amino acid residues across the membrane and quantify the insertion contribution from hydrophobicity and the positive inside rule. The membrane topology predictor will locate the membrane spanning segments and determine their orientations by a sequence search for minimum insertion energy by to drive the membrane insertions, the hydrophobic amino acid must make large contributions to insertion. And to orient the protein with respect to the membrane plane, positively charged residues must be strongly favored in the cytoplasm over the extracellular space. But some existing topology predictors have these kind of issues. They are statistic-based predictors. They cannot be used in targets that lack homology to previously characterized proteins. They cannot be used to design new proteins, and Levy scales would mean leader peptidase protein drives force of 0.5 kilocalorie mole in favor of arginine and lysine in the cytoplasm compared with the extracellular domain in lab measures. In this experiment, the authors developed an experimental method called deep sequencing toxcat beta-lactamase, which the hydrophobicity at the membrane core was in line with biophysical measures, report large asymmetries for the localization of the positively charged residues, arginine and lysine, in the cytoplasmic site relative to the extracellular site of the membrane. Thus, they developed a topology predictor called topgraph, which used deep sequencing toxcat beta-lactamase to assign insertion energies to segments of acquired proteins and search for the topology of minimum insertion energy. In this way, topgraph was tested on experimentally determined topology databases. Results are depicted in some graph. Letter A is the evaluation of the overlap related with membrane spans, and they evaluate the orientation prediction accuracy using 609 Escherichia coli inner membrane proteins with cytoplasmic or periplasmic location that was determined. They compared the performance of top graph and top cons, which is a topology predictor that used the consensus of 5 statistical predictors in these 3 tests. Letter B is the distribution of insertion energies in individual membrane spanning segments, and C is the experimentally determined structure annotated according to the top graph MSA prediction. This is a schematic representation of the graphical topology prediction algorithm top graph. Here we have a comparison between a predicted topology and a simulated topology. In A, we can see a case study demonstrating that the positive inside rule may favor membrane insertion of polar segments, and letter B represents a molecular structure annotated according to insertion prediction. TN7 in red and positive charge in turquoise. We can read here all items for the discussion. For our final discussion, we can say that top graph allows quantitatively examine aspects of membrane protein topology and may in the future help design altered or new membrane protein functional sites. May be used to test hypotheses on the insertion propensities of natural and engineered proteins and the deep sequences Toxcat beta lactamase scale is highly recommended as the implicit solvent term in structure prediction, design, and dynamics of membrane proteins.